For our next panel, we have the bioinformatics panel. Similar to the sample prep panel, this is all about the Q&A, so please jump in the Q&A and submit your questions for our bioinformatics experts. Much like the sample prep folks, these guys work within the genome generation world, just on the opposite end of the spectrum within the bioinformatics world. So I'll pass the mic. I know I need some help when it comes to bioinformatics. I'll pass the mic to some experts, Duncan Kilburn from Circulomics and Anthony Schmidt from Arima Genomics. Thank you, gents. Cheers.
Thanks, Christian. Yeah. Welcome, everybody, to the bioinformatics panel. My name is Anthony, not Joe, but that's okay. And I'll hand it over to Duncan to introduce himself and the session.
Okay, thank you, Anthony. My name is Duncan, and I'm going to be co-moderating the session. I work for Circulomics. Firstly, I'd like to thank in advance the panelists for joining us for what I'm sure will be an illuminating discussion. And we hope that we have all levels of experience in the audience, from beginners to experts. And so the session was planned with the aim of being able to get practical, hands-on advice and knowledge from experts in the field. So don't be shy, any level of question, please jump in and ask. So the session will be organized into two sections. Firstly, we're going to get the panel members to introduce themselves and just also briefly introduce an area of genome generation bioinformatics that they work in, including maybe what they see as important future developments in that particular field topic. And so we hope that these will prompt follow-on questions that we can use in the second section of this session. So please get those questions coming in. So firstly, I'd like to introduce Hugh Olsen from UC Santa Cruz. Okay, Hugh, please just give us a little introduction, and he's going to introduce a little bit about alignment and polishing.
You're on mute, Hugh.
Thank you. I work at UC Santa Cruz in the Genomics Institute, as well as the Department of Biomolecular Engineering in our nanopore sequencing lab. We're currently associated with, we're participating in Pan-Genome Project and T2T. I've been good fortune to work in a very talented group of bioinformaticians that include Benedict Patton as well as Karen Miga. And our group has been busy pioneering a new assembler called Shasta that some of you may be familiar with, that is based on principles designed to tune to the strengths of nanopore sequencing to produce an efficient production of an accurate assembly. And so I would be happy to try and field questions regarding alignment and assembly and polishing related to nanopore sequencing, because that's really the area that we've been working in.
Okay, thanks, Hugh. So second up, Sergey Koren from NHGRI. Please introduce yourself and just give us a little introduction to maybe assembly and scaffolding.
Sure. I'm Sergey Koren. I'm at the NIH. I've been at the NIH about 5 years. And we've really focused in our lab on building efficient tools for analyzing sequencing data. So some of the tools you might be familiar with, like MASH, WinnowMap, MASHMap, as well as genome assembly tools like Canoe and HiCanoe, which takes advantage of the new HiFi data that you heard about before earlier today from Karen Miga. And we've also obviously participated in the T2T project. And so as a quick overview of genome assembly, essentially it's a jigsaw puzzle, and it's easier to do the puzzle if you have bigger pieces. So the assembly algorithms all try to look for unique bits at the edges of these puzzle pieces. And if your puzzle is all sky, then there's nothing to put together. You don't know where to put the pieces. But if it has some unique features, like a bird on that piece of sky, then as long as the picture is high enough resolution, you can say, okay, this is unique, so I can put this piece of sky with a bird here and tie it together with the other piece that has a bird, and so on. And so that's how to think of a genome assembly. Scaffolding is a similar thing. You're trying to build on top of what you got from the initial result, except usually rather than having continuous pieces of sequence, you have bits of pieces that are separated by some known distance. And so you put together a puzzle, but now the puzzle has gaps that you know the sizes of, and so you know you should skip 2 or 3 spaces.
Okay, great. Thank you, Sergey. So next, Miten Jain from UC Santa Cruz. If you could introduce yourself and say a few words on variant calling.
Sure. Thank you, Duncan. Thank you, everyone. I'm Miten. I'm a research scientist at UC Santa Cruz in the nanopore group as well. And we are a group that works on developing solutions to complex questions that are interesting to our group at the time in biology. And then going with that, we develop software tools. So over the years, we've developed alignment tools, assembly tools that you heard from Hugh, and variant calling tools. So in the world of genomics, most of the times when you sequence a new human genome or any genome, you're not assembling a new genome, but you kind of know what species or what kind of genome you're assembling. And the question is how different it is from what is the known or conventionally referred to as the reference genome. So if you were to sequence human genomes, you're mostly going to ask the question, how different is it? And catalog those variations or single nucleotide variants, structural variants, and modification differences. And that's kind of where variant calling stems from, where you get a catalog of where differences lie and how they correlate. So there will be some variants that are common to specific ethnic groups or haplotypes and so on and so forth. And similar to that, we are developing some tools on variant calling pipelines in collaboration with Google Health, where the anticipation is that when done, we'll be able to move the variant calling timelines on a human genome level down to a few hours, essentially the goal being a few hours for assembly, followed by a few hours for variant calling.
Okay, great. Thanks, Miten. And next, Shilpa Garg from Harvard Medical School. You'll introduce yourself and talk a little bit about haplotype phasing.
Hello, everyone. I'm Shilpa Garg, a K99 research fellow at Harvard Medical School and Dana-Farber with George Church and Hangley. I'm passionate about developing new algorithms for complex biological problems. The main problem that I'm very interested in is haplotype phasing. So as Miten mentioned, you know the variants as well as you know their genotypes. But the next question we ask is how the alleles, they are co-located along the chromosome copy. And I'm excited to solve this problem using Hi-C method with innovative algorithms. And computationally, there are 2 ways to solve this problem. One is you take the scaffolds and do the Hi-C phasing in the space of scaffolds and then partition the reads. And this method, we have put up a preprint a couple of months ago that's up on bioRxiv. And the another way is to combine the phasing and the scaffolding step jointly into the graph space. And that these graphs can come from any local sequencing data information. And by merging the Hi-C based scaffolding and phasing, you can, for the first time, able to see chromosome scale diploid assembly, that you can essentially connect the chromosome arms. And having chromosome haplotypes is very important for biodiversity and clinical studies like long-range promoter enhancer interaction, as well as detecting mosaic cancer alterations. And our team is working in these directions. Thank you.
Okay, great. Thank you very much. Let's start out with some of the questions from the audience. So the first one here, do you have any, and I guess this can go to anyone who'd like to answer. Do you have any suggestions for polishing when more than one haplotype exists?
I think all of us can go, but given the most recent work, Serge has will have the right recommendation if I had to put him on the spot.
Right, so the answer is you should try to bin the reads as best as you can, whether that be with Hi-C data like Shilpa mentioned, whether that's with trio data if you have that available, or there's also an algorithm from Evan Eichler and others that uses strand-seq data to bin reads. But essentially the key is you don't want to give the polisher the wrong haplotype when you're polishing, or it will do very bad things.
ask a follow-up question on that? Actually, I had something about polishing as well.
Yeah.
Could maybe could you try to compare and contrast? I guess like given a certain input data, let's say it's ONT, what's the best polishing strategy? What data type do you use for polishing? How many rounds, and some of the details around that?
So I don't know if Miten, you have probably more recent experience.
With ONT, with nanopore, typically the variant calling frameworks have the classic route up until a year ago was essentially you would have the assembly from Canoe or Fly or Shasta, and you'll follow it up with a few rounds of Raycon and then followed by Medaka. We introduced a couple of polishing tools at the time, about a year ago, where one of them was margin polish, which is an HMM-based polisher that takes the output from the assembler and does a standard HMM error model-based polishing. And then we follow it up with Helen, which gives you a homopolymer correction as well as base correction, which is the further refinement on that polishing. And again, as Serge mentioned before, having a prior phased assembly makes a lot of difference to this polishing. More recently, there's of course more development on Medaka. That still is the Oxford Nanopore recommendation for this. We are in parallel developing a framework in collaboration with Google Health, where what we do is we run a candidate caller. So essentially we have a neural network named Pepper that goes into the assembly and picks out the variant calling candidates. And at this point, the assembly can be unphased. Once the candidates are there, we have two ways to phase those candidates given the assembly, using either something like WhatsApp or an internal tool that's under development called Margin. And with these phased variants, then we can run a round of deep variant, which is Google's variant caller, that gives you very high quality predictions on those variants given the haplotypes. And what we have seen in the recent Precision FDA challenge is that using the combination of Pepper and deep variant, the nanopore based variant calling is essentially superior to Illumina based variant calling.
So one thing I'll add on top of polishing is that we've sort of been moving away from polishing because of the hi-fi data. And so we've put a bunch of work into HiCanoe to make very good consensus straight from the assembler from a good quality data type. So I think as the ONT data improves as well in quality as it's doing all the time, we may get to the point where we don't really need to do polish anymore. So, you know, we've seen in the T2T project, there were only about 1,000 corrections that had to be made to a 3 gigabase genome. So you can imagine, you know, that's already like Q60 or something, and it took it up to Q70, but Q60 is pretty good. And doing naive polishing actually would made the assembly much worse because it you know, even though that genome was haploid, there were heterozygous variants in some regions, and those got unphased by the polishing process and all those kinds of issues. So it becomes quite difficult to polish some of these recent assemblies given their qualities.
Just adding to Sergey's comment, so another possibility is to correct the reads like in the initial stage, like when the general assemblers do either debridgen or overlap based strategies. So and also incorporate the phasing during the overlapping step and do correct the reads to these partitions. And this will reduce the errors in the subsequent consensus calling steps.
Okay, we have another audience question here. Is there any community interest in using Arima Hi-C 2.0 high coverage for high throughput de novo assembly of single end reads, followed by using the paired end Hi-C information for scaffolding? A one-stop shop possible, question mark.
I think we're moving away from short read assembly, so I'm not sure how useful that would be. So, you know, if it wasn't short reads, maybe, but with short read Illumina like data, I don't think there's that much utility given what the long read assemblies can produce.
Yeah, just long read assemblies can able to already produce the chromosome arms, and the centromeres in humans, they are quite big, like more than 30 megabases. And then to until we reach a point when we are able to assemble the centromeres, we still need some high-C data to connect the chromosome arms to produce chromosome scale assemblies. And this is where I think high-C is helpful.
What's more likely in the immediate term, to add to both of those comments, is that at the point, both nanopore long reads as well as hi-fi, people are still aiming for 30 to 40x coverage. What's possible is that the combination of improved lengths and higher accuracy could lower that down a little bit further. And then you combine that with high-C, and then that becomes the cost-effective high contiguity genome recipe.
Though I will say that hi-fi data, you can't aim much lower than 30x because one, it has uneven coverage. So there's regions it undersamples to some point, completely doesn't sample. And two, you're almost always assembling diploid genomes. So if you have 30x of the coverage, you really have 15x per haplotype. And so the combination of the fact that you have only 15x starting out and the fact that hi-fi has these dropout regions, you end up, with a good number of regions that are much lower than 15x. And so if you aim lower than that, you're going to systematically start having gaps of coverage, right? So at least with current tech, you can't necessarily go lower until those issues are fixed, even if you could scaffold it back together, but you'll end up with pretty big gap regions.
Okay, thanks everybody. The next question here is from Griette. It says, with short read sequencing, you would typically mask the reference genome to filter out dark regions. Do you still recommend to apply the same sort of masking for long read sequencing approaches?
I wouldn't.
Go ahead, sir.
I wouldn't mask. I mean, I would use a tool that's aware of the repeat structures, like WinowMap is specifically designed to map to repetitive regions. But I wouldn't mask, especially if you have, if you're mapping to a genome like CHM13, which has valid centromere assemblies and things like that, I would let WinowMap or GridMapper map to those regions too.
I agree. That would be it partly would defeat the purpose of the long read if you were to mask the repetitive regions. It's handling the data rather than masking.
The other part is there's a random. It's some level it's random, but we have reads that go into the millions of bases, and so what you want to see if you can map into those regions and with those long reads. And as we continue to build higher and higher percentages of these incredibly long reads, we're hoping that becomes more useful. So it seems like masking would not help with that anyway.
Okay, thank you. So next question here is from Sarah Frampton. Sniffle's GitHub page suggests it performs best with mappings from NGMLR, but in Karen's talk, I saw they've been using WinowMap 2 for alignment. Is there an advantage to this?
So I'll answer that since WinowMap came from our group. So Chirag, who is the primary developer, has done a bunch of testing comparing sniffles on inputs from NGMLR, Minimap2, and WinowMap, and he's found that it has better precision and sensitivity than both of those other options, especially in repetitive regions. So we expect we'll have a preprint about that in the next couple of weeks. But yeah, I think the Sniffles page is posting it for NGMLR because the page was written before WinowMap existed.
Okay. The next question here is also for Sergey. Can you give more details about the hi-fi dropouts?
Sure. So primarily we've seen it in large stretches of GA or CT, which is the reverse complement version. And the way I think of it in my head, my model of what's happening is that this DNA is forming a secondary structure, which makes the polymerase either slow down a lot or even maybe drop off the gain of molecule. And the longer the sequence of GAs is, the higher the probability of the polymerase either stalling completely or falling off is. And so if you think about what a hi-fi read is, it's a 20 kb read that you go around 10 times, right? And so if your polymerase is super slow, you'll never make it around that more than once, or if it falls off or completely stalls, right? And so when you try to make a circular consensus read, you're only going to have one pass, which you'll never get to Q20 or the minimum threshold that hi-fi reads require. And so you'll end up with no read there and no data there. You can get a CLR read there, but you won't get a CCS read. And if this GA stretch is not too long, then maybe the polymerase will make it around just very slowly, and you'll get some passes. It might be a little bit lower quality, but you'll at least have some data, but you won't have, the normal coverage. It'll still drop down because some fraction of polymerase has never made it, right? And so we've seen this. We've looked at it most carefully for the CHM13 data, and so I can say there's about 30 of these regions in the human genome where the hi-fi data is completely missing. It is much worse on some other genomes, like in some of the fish that we've looked at with VGP. You can actually see the bias is so severe that you can see in the Cambridge distribution a shift to lower coverages. So instead of having a peak of coverage at 30x, you have a smear of coverage from like 20x to 30x and down, because there's K-mers that should have been 30-fold, but now they're only 20 because they're next door to these GA tracks, which are knocking out your polymerase and knocking out your sequencing reads.
Sergey, follow up on that. Do people look at the CLR equivalents of those reads, and are those signals any use if you're looking at these specific narrowed regions?
Yeah, so I looked at that a lot in CHM13. So we actually took so we got the data where every ZMW produced a read, essentially was what we asked for. So they are essentially CLR reads. I never found a read for any of those really long J stretch regions where there was more than a single pass of a read. But they can certainly be useful in that you can use them to gap fill a scaffold and things like that. So I think the currently recommended strategy I take is you assemble the hi-fi data scaffold, and then you could use all the data, including the CLR, that couldn't be turned into high quality, but at least you can map them there. And if you have a few of them, call a consensus that's to fill that gap that otherwise you would just have no sequence for.
Okay, thanks. I have a question here from Isa Shern. What would be your suggestions for hybrid assemblies from ONT and fragmented Illumina draft genomes? She says we have no PacBio or good quality Illumina genomes yet.
I'd throw out the Illumina data and assemble the ONT data. And then maybe use the Illumina to polish. I
was going to say that.
Polish.
Generally speaking, at least this is almost 2 years old, but we did a C. elegans genome assembly project where you can also essentially see that not just in terms of quality, but also the contiguity, the nanopore only assembly is superior to nanopore Illumina, like a Spades hybrid, or any hybrid assembler for that matter. So your best bet is to do what Serge said. Use nanopore for assembly, polish with Illumina. And then just be careful with polishing. For example, Pylon, again, depending on how many rounds you use, it can start introducing artifacts after a point. So be careful when polishing.
There's one thing to note here that may have been brought up, but ONT came out with a very recent announcement and improvement in their base caller, in which they're talking about somewhere in the neighborhood of 97, 98% accuracy. I would say that the field is moving quickly now, and that there are large groups of very focused and able bioinformaticians. Sergey is a great example, working on this. And so if you have a project where you need to do an assembly, it is well worth revisiting it in 6 months, because things are moving very quickly, in my opinion. I don't know, what do you guys think?
Yeah, and also it depends on the genome that you are assembling. Like in case, like it's not a human genome and it's a simple fish genome, which is not very repetitive, like combining Illumina and like a hybrid, you can easily build a graph from Illumina and thread the ONT reads, because Illumina can give good information on the heterozygous sites, whereas it's a little difficult to call heads from ONT. But yeah, if you are doing the human, like large scale genome, very repetitive, like going via ONT approach and going to Illumina would be better.
Okay. Question here for the panelists. Does anyone have experience assembling plant genomes, and can you speak to the unique bioinformatics challenges with plant genome assembly?
I would not to again put him on the spot, but he's done more than we have. We have done a couple of small plant genome size projects, but Serge would be the person who's done across multiple technologies, I think, for plants.
Yeah, I mean, the hardest part of plants, well, there's a couple of things of plants. One is they're often, polyploid, tetraploid, whatever you may have, not just diploid, like most of the nice, well-behaved VGP samples or vertebrates, whatever you're working with. They can also be very repetitive, and they can have extremely large genome sizes, which makes the generating sufficient coverage quite daunting, potentially. I will say that I've been very happy with how hi-fi assemblies of plants have come out. So I'm aware of assembling the redwood genome, which is a 50 gigabase genome. I know some companies have done oat genomes, which are 20 gig, and other similar plants, and have gotten multi-megabase, like 20, 30 megabase counting and 50s, and quite good quality and accurate assemblies. Same goes for Koren, and similar genomes like that from hi-fi have been very good. And in the cases of polyploid genomes, you actually get all the haplotypes separated out. Your assembly is just much bigger than you would expect, and it's up to you then to sort them out and separate. Here's haplotype 1, 2, 3, 4, whatever you may have. But at least you'll get all of them separated much more than you would have in the past. So the computational expense of the hi-fi assemblies is not that bad. The data generation aspect is still there. So, I mean, you need to generate a lot of hi-fi data to get enough coverage of a 50 gig genome.
Okay, we had this question earlier, but I think it was suggested that we bring it back for this session. So it was from Rasmus Kirkgaard. At what error rate would nanopore ultra-long reads be enough to assemble diploid genomes separately? With nanopore starting to approach Q20, so the bonito that we just talked about. It would be interesting to know how much better it has to be for contiguity. Then it might be possible to use PacBio hi-fi reads to polish the contigs, ?
I think this is something we've been arguing inside as to where do we have to go to in order to get it where nanopore is sufficient or mostly sufficient to get you to telomere to telomere with Q50 or better. And honestly, I don't know the answer to that. How about you, Miten?
I think what Serge responded to Rasmus was very much spot on, in that the happy coincidence, along with the improvements in accuracy with hi-fi that happened with Karen and Serge's CHM13 work, was the heterogeneity of the human genome. Because of the haplotype diversity and heterogeneity being less enough or low enough that you could use those lengths and that accuracy to accurately assemble the genome, that made a lot of difference in terms of where the accuracy and length combination lies. It's kind of one of those uphill, but at the same time empirical battles, in that if you had X number of long reads and the errors are low enough, can you move from one paradigm to the other? Likely the answer is yes, but we don't know. I mean, at least from a long read development standpoint, we have placed the bet on that ultimately the longer reads will end up being the superior workhorse in that. But some of that, of course, is contextual, in that all of this likely holds true, and Serge should comment on that. With human genomes, there's obviously use cases and variations to occur once we get past the conventional human genome paradigm.
In addition to error rate, I think the error modality also matters. For example, the errors are random or non-random. So one of the beauty of PacBio that we could able to resolve the diploid is because the errors are random there. So on a side, the probability of having an error in the reads is very low, whereas it might be a little higher if you have the non-random errors. So it's a function of both error rates and like the type of errors.
Devil's in the details for sure.
Yeah, and I mean, I'd add to that too, that a lot of these stats, about identity, I'm skeptical that they will apply across all genomes, right? So I mean, Nanopore is trained on sequencing context, and it's hard to learn all the sequencing context from just a handful of genomes in the training set. And so while it may be 97, 98% on a human, is it going to be 97, 98% on a random amphibian or some other genome we've never seen before, right? So there's some caveats that follows up on what Shilpa is saying, that, it matters not just the modal error rate and modal error rate on a human, but also the type of errors and whether they are higher in some genomes than others and so on. I think, keep in mind, so the Q20 kind of is a threshold of hi-fi. That's the worst hi-fi read that you would keep. In practice, when you homopolymer compress them and correct them like we do for assembly, we're assembling hi-fi reads at tolerating no errors, right? So that's how we built the CHM13 graphs that Koren showed. That's how we resolved the human to be 6 gig. And so you need to get below that 1 in 1,000 or so for human error rate to be able to confidently say this is a variant and not an error in my read. Now you can miss some of those if your read is long enough, right? But still, I think there's a good distance to go before you're able to do that so easily with the ONT reads as you can right now with hi-fi. That doesn't mean that ONT won't get there. But I think even Q20 is not quite there yet.
Right. Well, and the other part I worry about is, to your point, is our sophistication and understanding structure comes up. And the training to specific data sets like the human genome, that the error model for ONT's base caller will improve. And that's absolutely critical. I agree with you that there may be aspects, fine detail aspects of that no one appreciates yet that are really critical to getting to where we need to be. So.
All right, I'm gonna do just a time check. I think we're all out of time for this session. I wanted to thank all of our panelists very much and everyone for their participation. There's a couple questions that we didn't get to. I don't know if the panelists wouldn't mind following up to anonymous attendee and Sarah Frampton about their questions. And thank you very much again. And then I'll hand it back over to Christian to give us a time update for the next session.